Do you want to start with a quick introduction about yourself, like who you are, where you went to undergrad, grad, doctorate, and then where you're at now?
Let's see, I grew up in Florida, in Central Florida, and I went to college. Well, I went to high school in Daytona Beach College, and then I went to college in at Stetson University. And so I was there, of course, 4 years. And then I wanted to go to grad school in oceanography. At that time, oceanography was a big, exciting field, and Jacques Cousteau was people very excited on television. And so I went to Scripps Institution of Oceanography, which is in San Diego, actually in La Jolla, California. So
Yeah.
I traveled all the way across the country there and started grad school there. And then I went from there to doing, after I got my degree, to doing 3 postdoctoral periods. One was back in Boston at Harvard, another at Woods Hole Oceanographic Institution down in Woods Hole on the Cape Cod. And then finally at UCLA, I did my last one. And then from there, I got a job at the University of Southern California in the biology department. And there, got tenure there. I was there about 15 years, went to University of Hawaii. I was recruited out there, was there for a while, recruited to University of Wisconsin-Madison, was there for about 10 years, back to Hawaii for another 10 years, and then now I'm at Caltech for the last 2 years.
I find it interesting that you did 3 postdocs. Was there a reason behind that, or?
Yeah, there were sort of 2 reasons behind it. Back in those days, postdocs were relatively short. Maybe 2 years was probably the average. And I had decided I was excited about becoming a microbiologist, and I had no microbiology as an undergraduate or really even as a graduate student, except I worked in the lab of a professor who was a microbiologist. So I learned a lot from him. But I realized I needed to have a little more experience in the area of microbiology if I wanted to make that my career path. So each of those 3 postdocs I did, I think it was 2 years, 2 years, and 1.5 years. So each of them were not very long. It was only a total of 5.5 years. And there are a lot of people who do postdocs now for 4 or 5 years in one place. So back then it wasn't a big deal to do that, to do postdocs if you were doing short ones like that. But it was mostly to get a background in microbiology. I worked with 3 outstanding microbiologists and really felt like I was ready to move into my own position by that point.
Yeah, that's really cool that you did 3. And I think, do you mind talking a little bit more about your own research in understandable terms for a high schooler?
Yeah, sure. It's a really fun area, and I think it's accessible to most people. I think everybody knows that the microbiome, the bacteria and other microbes that are associated with animals, plants, are very, very important to their health. We have sort of rediscovered that in the last 10 years, and people have gotten very excited about learning more about how that interaction is between a bacterium and its host. And in particular, how do you set up that arrangement at each generation? Because many animals and plants are like humans in that we're essentially sterile when we're born, and we have to now basically make friends with a bunch of microbes that are going to be lifelong companions and going to help us in so many ways. The problem is the human microbiome and most microbiomes of animals and plants are very complex. Like ours is hundreds of species in the gut tract alone, and it's very hard to understand what the communication is when you've got a crowd that big. It's like going to some sort of rave and you're trying to understand a conversation in there with all the other noise going on around you. So we chose a symbiosis that was a simple one between a single bacterial species and its host. And in this case, it's a luminous light-emitting bacterium that is cultured in a light-emitting organ by a marine squid. And there are a number of marine organisms that use bacteria as their source of bioluminescence. So we're very interested in how that relationship gets started in the marine environment. For instance, in this squid, it hatches out of its egg without any bacteria. There are, millions of bacteria in the water around it, many hundreds of species, and it's able to pick out a single species from all of those that are in the seawater and allow only that one to come inside its body, get into this light-emitting organ, or this organ which will, once the bacteria are growing and emitting light, will be used as a sort of a flashlight by the host, by the squid. So basically what we're interested in is how do you recognize your specific symbiont amongst all the other bacteria that are present? It's sort of like I don't know, the old spy movies where you'd have a password and there'd be a counter password, and then you'd have a counter password. And imagine doing that 10 times, 15 times to be absolutely sure that the person you're talking to is who you think they are. That's the sort of process that an animal needs to go through to make sure it's got the right bacterium before it lets it come in and grow inside of its tissues. Because that's a really big decision to make, to let bacteria set up house inside your organ.
Yeah, and then I'm curious, what is your like methodology, like data collection process like?
There's several ways we do this work at a bunch of different levels. The simplest is physiology. We just look at the appearance of light by the squid after. Oh, okay. I guess I should back up. We have an animal model. It's this little squid. You can bring them into the laboratory as adults. They lay eggs. Females lay eggs. The eggs hatch, and out come these little juvenile animals. They're about the size of a fruit fly, and they will immediately begin trying to find this bacterium that they want. And so we can provide to them in the water either none of that species of bacteria, in which case we watch them be able to keep all the other bacteria out of this nascent organ, or we can give them whatever kind of bacterium of the species that it will take up. And by kind, I mean we can make mutations in that bacterium in that particular species, in a strain of that species, and see whether or not loss of one gene or another, or gaining of genes, how that changes the ability of the animal to recognize and allow the bacterium to set up shop inside it. That's a very simple thing to assay because the animal gives you a reading whether or not it was a successful symbiosis by just producing light. And so you can put the little animal inside of a vial, put it inside of a photometer, which measures light production. And if there's light being produced by the squid, that means it was a successful colonization. If there isn't, then that means that that particular strain or mutant was unable to colonize. So that's our sort of simplest, most straightforward thing we do. But we also do a lot of omics, that I'm sure people have heard that term. It's like transcriptomics and proteomics, genomics. It's basically comparing between experiments or between samples, either what the DNA sequence looks like, what the RNA sequences are that are produced, or what the proteins are that are being produced during that relationship or that conversation that's being had between two species. Those are two of the things. And the final one I'll say is imaging. Imaging is a hugely important way of understanding what's going on when two different organisms are interacting, especially microorganism and a host tissue. Once again, imaging kind of went into decline about 25 years ago, and people thought, well, it's kind of old stuff. Microscopy sounds old. And so, and then a bunch of really new technologies came on that really made it possible to go deeper and deeper and deeper into what these images look like. And we discovered all kinds of amazing structures and interactions that we hadn't known were there before. So imaging now is a very, very important of our research program.
Which is that you mentioned that there's like a research and a lot of research that lost interest. And I think the big one that's hit the public is your gut biome, and where do you see the field going in the future?
If I knew where it was going, I could go there.
Yeah.
No, research, you don't really know where things are going. You just ask the best question you can at the time and hope that opens up doors that then make it clear where the next step is. However, yeah, you're absolutely right. The microbiome is a very big deal right now. I think what I would say is going to be the future is personalized microbiome work. In other words, I'm sure people have heard of personalized medicine. In other words, we don't give everybody, 2 aspirin and tell them to, go home. It depends on what your particular manifestation of a disease, a certain condition is, and depending on your genetics and your physiology and your, who you are. And so I think the same thing's going to happen with the microbiome. I mean, there's no microbiome. There are millions of microbiomes. You have one, I have one. You have one on your skin. I have one on my stomach. I have one on my skin. I have one, So there are all these different microbiomes, and getting to know them and what they're doing for us will help us design personally what I need when something goes wrong in my gut tract, or something goes wrong in my skin, or in areas of the body that microbes are such good helpers to us. So that would be the area there. As far as CRISPR goes, yeah, CRISPR has just opened up. Basically, what CRISPR has done is opened up genetics to almost any animal, any organism, plant or animal, actually, that you want to genetically modify. It used to be there were only a few kind of animals you could genetically modify in the laboratory. There were only a few, even only a few bacteria you could genetically modify. Now there's maybe, 100 that you could modify in the ways that we developed over the last 20 years. But with CRISPR, you literally can do anything, anytime. And so it's crazy. It's like some sort of science fiction thing where, you have this magic instrument that allows you to do something that, nobody used to be able to do before.
Yeah.
So it's a technology. It's an important technology, and it's going to make a great deal of influence on people and their health.
How did you find your passion, and how do you suggest high schoolers find their passion?
I found my passion with a biology teacher that I had in high school. I can go right back to that moment in that laboratory and that he would do, and he did all these things that nobody else in biology classes in high school were doing. He went out of his way, bringing materials that only he could get his hands on, or was an effort to get his hands on, and opened up a lot of doors for us to try things out. Just watching his enthusiasm at doing that, and this was not a young guy either. This was this was a guy who'd been teaching biology for 20, 25 years, and he was still super excited. So I think passion can be internal. It can be something that you kind of know for your whole life. That's what you want to do, or it can be infectious. You can pick it up from the people that are around you. And in my case, I think that was it. He certainly sparked it in me. So yeah, I would look to your teachers as one source. Of course, other people that you meet in your life are going to help you develop your passions, whatever they might be.
What type of skills can high schoolers start to develop now? Like even if they don't have access to a wet lab, what steps can they take to get knowledgeable or into research?
That's a very good question. And I've got probably some unconventional answers. If you want to go into science in particular, the most important class you will take in high school is English. And by that, I mean composition, learning how to write a book report, learning how to write an essay. It turns out that all scientists, after they get a job, they realize that's the one thing they need to do the most, is because it's not enough just to do something. You've got to tell people about that. There's two ways to do that. You can talk to them about it. You can present seminars and whatnot. And that's also a good skill to learn is public speaking. A lot of us are not very good at that and never got any training in it. And we had to learn it kind of the hard way. But really what every day you're doing is you're writing up your results, you're writing a report, you're eventually writing manuscripts that are published in journals. Writing is incredibly important. And so that's the unexpected answer I'm going to give you, is that I think some of the most important courses for a scientist to take in high school are the compositional courses. And I had a fantastic English teacher, and she was hard as nails, and she really made us write. And it's so important. The second thing I'll say really quickly is that I think learning to ask questions, putting your hand up whenever somebody is talking to you, whether it's in a class or seminar or even just one-on-one, be thinking about what's the next question? What are you interested in? How can you find out more about what this person is saying? And have the guts to do it. Start it in high school, because the sooner you start doing that, the safer the environment is. In other words, there are people that and it doesn't matter. You're not going to lose your job if you if you ask a question that you later on wish you hadn't. Just try it out, do it. So anyway, that be an inquisitive mind, because everybody likes an inquisitive mind.
If you were to hire a high schooler, right now, what type of traits would you be looking for? Like, what is that trait that makes you say, I want them?
Yeah. We have high schoolers in the lab almost all the time. One is, I think the most important one is this enthusiasm. Science is something that you do. Maybe you work for several days on something before you get a positive feedback, before you get an answer, or before you get a, pat on the back or something. You feel like you've done something and are really excited. And so you have to have enough enthusiasm and enough excitement about what you're doing to get you through, a few days that are kind of either boring or maybe even disappointing because things don't work. So you kind of look for somebody who's got that kind of stamina, if you want to call it that, sort of intellectual stamina. Also someone who's communicative, like I was saying earlier. Well, I mean, everybody knows it's a lot easier to work with somebody who will talk back to you. Dedication and actually the ability to put the time in, and reliability. In other words, they say they're going to be able to put in 10 hours a week. You really want to know that they've thought that through and they're able to do that. And they make a point of doing that. So those, I think, are kind of key personal traits. And it really doesn't matter. I think that goes for almost anything. It doesn't matter whether you're a scientist or in a microbiology lab or whatever you do.
Thank you so much for your time today.